Hey everybody, Mr. Bremberger back. We're going to talk about incomplete dominance as well as codominance, multiple alleles, and some sex-linked traits. We've got a full package of information here for you today to go over some of the not so nice working out things of genetics. I've got so much information here for you that I had to actually get rid of my face from the screen to fit it all in. So I hope you guys don't mind me not being in the videos this time around. Now, we're going to talk first about incomplete dominance. Now, in incomplete dominance, neither allele is going to dominate. So there really ends up being kind of a blending of those traits when the two different alleles for the same trait occur together. So colors will tend to blend, which leaves that heterozygous kind of equaling a third phenotype. If you take a look at the flowers that we have in the image at the lower right hand corner, if you take a yellow and a white, put them together with the with the heterozygous genotypes, we can end up getting the pink, which is the blended combo. If you take a look at this via the Punnett square with your red genotype and phenotype, RR for red, and you cross it with the pure white, which here would be WW. Again, although you're dealing with different types of alleles here, we're still concerned about the same gene. Okay, so we can have different combinations of letters occurring here for your particular type of genotype. You don't necessarily need to use the same letters, although if you wanted to call the white purebred, in this case lowercase r, you could. And then just understand that anytime you get the heterozygous genotype, you're going to end up with a pink flower. So that is the beauty of incomplete dominance. Codominance is when you're going to be able to see both of those phenotypes present in one particular organism. Again, they're showing up simultaneously. So in cattle and horses, let's say for example you cross a pure red horse with a pure white horse. No, you're not going to get a pink horse, but rather you're going to get a type of color known as roan. So red and white are going to equal roan. The same thing will actually happen with cattle. Here is your roan phenotype illustrated in the picture in the lower right hand corner. So again, this is I guess we could bring up a different example, and that being what would happen if you crossed a roan with a white. To show the Punnett square, you take your RW, which would be your red white roan phenotype, with the purebred white. If you go to complete the square, we're going to end up with a roan and a white across the top, and also a roan and a white on the side, giving us kind of a 50-50 shot at either achieving a roan cattle or a white one. Also, we have multiple alleles that can be displayed in different times. So, for example, in this pattern, the genes are going to have more than two alleles controlling them, and are therefore said to have multiple alleles. Now, no individual has more than two alleles, but there are more than two possible alleles in a population. So that trait can be expressed in more than two forms. A lot of talk. Here's an example to hopefully make this make sense. In the classic one is that of blood type, right, in humans. So, for example, humans are going to have generally four different blood groups. You've got A, B, O, as well as AB. Now, again, these are produced by three different alleles. So we've got four different groups produced by three different alleles. Again, those alleles are A, B, and O. And again, there are your combinations. If you take a look at the right, here's your phenotype, A, B, and O. Genotypes, as they shake out, in order to have a type A blood type, you're either going to have AA that you get from your parents, or A and O. All right. Same thing with the B blood type. AB, you got to get both AB from mom and dad. All right, or A from mom, B from dad. And finally, you've got to. I'm sorry. Yeah, let me back that up a little bit. You know, if you're going to be AB, you got to get A and B from mom and dad. O would be O and O only. No A's, no B's, or you know, AB. You can only get an O from both parents. Here's a Punnett square. Check it out. We start crossing these things. We take an AB parent with a type A parent that is kind of heterozygous, I guess you could say, for that particular genotype. And probability-wise, 25% are going to come out AB, 25% B, and half of them will be type A. All right, if you take a look at, you know, your heterozygous AB blood types or A cross B blood types at the right, you can, you know, take a look at those results and analyze those if you would like. And no, by the way, we are not going to be blood typing here in class. That is something that we just don't mess with anymore in schools because of those bloodborne pathogens that can be easily transmitted from one person to another. If you're interested in the blood groups specifically, you can take a look at these. And that is the end of our show. One thing that I did not mention here is, at least in the interest of time, is the concept of sex-linked traits. So what I'm going to do is add another video for you talking specifically about sex-linked traits.